Blakely, super, thank you. I'm Helen McShane and I'm the director of the Validate Network, and I'm really absolutely delighted to introduce our two speakers today. So first to introduce Professor Samantha Sampson. Sam is at Stellenbosch University in South Africa, and she has a research group focused on mycobacterial genetics, microbiology, immunology and animal models of TB. Sam holds a South African Research Council chair in Mycobactomics. And this is really, this chair has really allowed her to establish a research group focusing on TB host pathogen interactions. And then Jomien Mouton, I'm equally delighted to introduce, is a postdoctoral scientist working with Sam in her group at Stellenbosch, and Jomien is also one of our two Validate fellows. So really delighted to introduce you both and very much looking forward to the seminar. Thank you. I think maybe from the slide Jomien is going to start, but I'm not sure. Over to you. Jomien, are we starting with Jomien or are we starting with Sam? It looks like Jomien.
With Jomien, she's still on mute.
Apologies. Yes, I was still on mute.
Lovely.
Thank you very much for that kind introduction. Can everybody see my slides and hear me?
I can, so I think it should be all right.
Great. Yes, so thank you, Helen. To start off with, as most of you are aware, NTB, the causative agent for tuberculosis, exhibits a biphasic kill curve where initial rapid killing is followed by much slower killing. And these slow to kill bacteria represent a population of persisters that are known to be reversibly drug tolerant but not genetically resistant, and are characterized by slow or absence of growth. It is thought that these persisters underlie latent infection, and latent TB infection is a clinical state where individuals are infected with TB but they don't present with any clinical symptoms of disease. Unfortunately, persisters requires lengthy antibiotic treatment and poses a risk for reactivation, which causes regrowth or reactivation and causes active disease. The exact mechanism which is responsible for the exact mechanism that is responsible for MTB altering its replication dynamics in response to host cues are not understood. And it is possible or likely that inherent properties of these bacteria may render them slow to kill in general. Therefore, studying and finding and isolating MTB persisters to be able to study them to improve our understanding of these bacterial populations seems like the ultimate quest. It's a bit like finding a needle in a haystack. But as we like to view it, persistent, with a little bit of persistence and the right tools, we are able to dig a little bit deeper into the understanding or our understanding of these bacterial populations. In order to overcome this specific challenge, we developed a dual fluorescence replication reporter that is called Fluorescence Dilution. And this tool provides us with the opportunity to monitor bacterial replication at a single cell level. So this tool makes use of two fluorescent reporters, one GFP that is constitutively expressed, and serves as a marker for bacterial viability. And the other, a far red reporter, is under control of an inducible promoter. So in a bacterial culture, when the inducer is present, the bacteria will express both red and green fluorescence. In an actively growing culture, when we remove the inducer, we can observe a dilution in this red fluorescence intensity with each cell division. Subsequently, those cells that retain their red fluorescence intensity can be monitored using flow cytometry or confocal microscopy. We are also able, we've previously shown that we can calculate the number of generations using this tool, and that this compares really well with generations calculated from optical density and C-few. So when we apply this tool in vivo, we are able to infect host macrophages with MTB that are carrying the Deal reporter plasmid. Infected macrophages are allowed to infect for 3 hours before we remove the extracellular bacteria using penicillin and streptomycin. All of this happens in the presence of the inducer, and after this point we remove this inducer, and then we allow the bacteria to replicate in the absence of the inducer, and then we can track this intracellular bacterial replication using flow cytometry, microscopy, and colony forming units. So when we analyze this flow cytometry data, we use the following gating strategy. I realize that this is very oversimplified, but just to give you an idea of how we look at this. So we select our bacterial population of interest based on the side scatter and forward scatter. Within this bacterial population of interest, we then look at the red and green fluorescence. We select on a population that is high in GFP because we're interested in the viable bacterial population. Within this population, we can then assess the red fluorescence intensity over time and changes in this intensity, but also look at cells that retain their red fluorescence. So when we infected macrophages using MTB into it's a raw macrophage cell line here, we have observed the following results using flow cytometry. So on the x-axis, we have the red fluorescence intensity, and on the y-axis, we've got the bacterial number. So here, the arrow points to the high red fluorescence intensity. We can see here, and this represents intracellular bacteria at 0 hours. The black dotted line represents in vitro cultured bacteria at after 4 days of growth, and we can clearly see a shift in that red fluorescence intensity that is indicative of bacterial growth. After 1 day inside the macrophages, bacteria are starting to represent this bacterial heterogeneity that we see here, which is even more emphasized after a longer time being exposed to the host environment. And here at the bottom, where, I don't know if you can see my cursor, but at the bottom right corner of this orange peak, we can see a bacterial population that retains its high red fluorescence intensity, representing a population that is either very slow in growth or non-growing. So when we looked at confocal microscopy of these samples, and I would like to point your attention to 96 hours here, we observed several bacteria in the macrophage that is green, so they are all viable, but only 2 of these retain their high red fluorescence intensity. So the confocal microscopy did not only confirm the heterogeneity that we observed on the flow cytometry, but also that some of these cells retain their high red fluorescence intensity and are therefore not replicating. So in a related study, we decided to apply this fluorescence dilution tool. And this is work that has been performed in our department by another research group, where they've found, persisting PET-CT lesion activity and MTB mRNA present in samples of these patients after TB treatment. For this study, 131 culture positive patients with pulmonary TB was recruited, and for these patients, PET-CT scans was performed for 99 of these patients at diagnosis, but also 1 month and 6 months after the start of treatment. So after treatment, these patients were classified into 3 different groups based on their PET-CT imaging. So cured patients demonstrated no lesion activity, failed patients demonstrated intensified lesion activity, and recurrent patients demonstrated new lesion activity. In addition, the study demonstrated using qPCR analysis of mRNA from sputum after month 6 after treatment and 1 year, which confirmed the presence of MTB in many of these samples. So to us, this remaining lesion activity and the presence of MTB in patients after treatment demonstrated the possible presence of persisters. We therefore hypothesized that a genetic component predisposes bacterial samples from the recurrent and failed group to have a greater potential to form persisters than those from the cured group. In order to test this, we selected 15 clinical isolates from this catalysis study, of which almost half belong to cured group and the other in the recurrent and failed groups. Because the sample sizes or the samples that we selected from the recurrent and failed groups were relatively small, we decided to group that into one group. Just a note that all of these samples were drug susceptible. These were clinical isolates from these patients. So the main aim of the study was then to identify the correlation between possible persister formation and clinical outcome in pulmonary TB patients. In order to do that, we wanted to look at this on a phenotypic level by making use of our dual reporter tool in combination with flow cytometry, and look at the whole genome sequencing of these patients at baseline and 6 months after treatment. So for the phenotypic persister analysis, these clinical isolates were transformed with the dual reporter plasmid, and isolates that contained this plasmid were infected into THP1 human macrophages. The macrophage infections were performed as described earlier, and we were able to track the intracellular bacteria of these isolates using flow cytometry from day 0 up until day 5. So firstly, we confirmed that the isolates are expressing both GFP and turbo using a plate reader, and then we determined whether the reporter had an effect on MTB growth. And here, these are just 2 representative images of growth curves from these samples, from the different groups. And in blue, we can see the wild type isolate, and in red, we can see the isolate carrying the dual reporter. And it was clear that the presence of the reporter did not affect the in vitro growth. So in the flow cytometry analysis, we wanted to assess the presence of or using flow cytometry, the presence of the slow and non-growing persister population. So this is flow cytometry plots. Again, on the x-axis, we've got turbo signal, and on the y-axis, the bacterial number. And the left plot represents a patient from the cured group, and the right-hand plot represents a patient from the recurrent group. The red line represents intracellular bacteria at 0 hours, and here we can see a high red fluorescence intensity because, yeah, this is what we expect at that time point, just after being treated with the theophylline. And the black dotted line represents in vitro cultured bacteria. So these bacteria have been growing for 5 days in vitro, and we can see a clear shift in that red fluorescence intensity. The orange lines represents intracellular bacteria after 5 days in the macrophages. So the blue line here, the arrow, points to a clear non or slow growing persister population in the recurrent and failed group, especially when we compare this to the in vitro cultured bacteria that's been growing for the same time. This is not that clear in the cured group, although we see some slowed replication in this orange population. It's not very much different to the in vitro cultured bacteria. These purple arrows indicate heterogeneity that was observed in the recurrent and failed group after time in the macrophages, and this is significantly more than what was observed in the cured samples. This black dotted line and the arrow indicates that we observed slower intracellular growth in the recurrent and failed groups in general. So if we measure the change in the red fluorescence intensity from the mean fluorescence of this red population here, we see that that was much less in the recurrent and failed group compared to what we observed in the cured patients. So when we looked at the percentage of persisters that was present in these macrophages compared to the in vitro cultured samples between the failed and recurrent group and the cured group, we observed a significantly increased percentage of persisters in the failed and recurrent patients compared to the cured patients. So for the genotypic analysis, we wanted to determine whether sequence variation predisposes persister formation in clinical isolates that was taken at baseline from patient groups. For this analysis, we performed DNA extraction using CTAB method. Sequences were subjected to Illumina sequencing using the MySeq platform. And an automated data analysis pipeline was used. So this pipeline is established in our department and it's been developed by the TB Genomics group. And after this, online software was used to predict the impact of SNPs and insertions and deletions. So this is just a very brief overview of the automated data analysis pipeline that was used for this analysis. FastQ and Trimmomatic was used for quality assessment and pre-processing of the sequences. Combination of 3 different alignment tools was used to align the sequence to the reference genome, which was in this case the Mycobacterium tuberculosis H37RV. Oops, sorry about that. And a combination of SAM tools and GATK was used to validate the alignment and also improve alignment. GATK was also used for variant calling and to identify the lesions using the genome coverage approach. So in terms of, at the time of the analysis for the NGS sequencing, only 9 of these isolates were sequenced, of which 5 were from the cured group and 4 were from the recurrent group. And we therefore decided to follow a comparative analysis where we compare sequences from baseline with those at month 6, because we had the data available for that. Sequence analysis, or the sequence, proved to have an average depth of coverage of 100 times and have had average percentage map reads of more than 90%. And this just indicated that the quality of the sequence was good. And interestingly, from this comparative analysis, we only observed variance in the recurrent and failed patient groups. So just a closer look at this analysis demonstrated that 5 of the 6 variants identified in this recurrent or failed patients were heterogeneous. So they were present only in a proportion of the population, either before or after the treatment, and only 1 of them were fixed. Of these, 4 were shown to have a non-synonymous amino acid change and were predicted to have a deleterious effect on the protein function by using Provian. Quite a lot of different genes and regulatory modules have been implicated in this analysis, but I would specifically like to focus your attention on the PE-PGRS18. Previous work has shown that this promotes the intracellular survival of M. smegmatis, which is a non-pathogenic model organism for NTB, by altering the macrophage and the cytokine profile and attenuating the cell apoptosis. And I thought this is especially interesting in light of Sam following me up with a talk about PE/PPEs. The follow-up, the variants in the follow-up group were not as many as expected, and this pointed to, the little variation pointed to little evolution during this time of treatment. So in summary, We identified a slow-growing population in isolates that's associated with unresolved or new lesions on PET-CT scans, which point to strains that are more likely to form persisters. On a genotypic level, we identified sequence variants in isolates from the failed and recurrent patient groups, and variants were located in genes that are associated with increased survival in macrophages. This work indicates the involvement of persisters or a slow or non-growing population as likely persisters and the inherent propensity for strains to develop persisters when exposed to host environments. And I think this is especially valuable information to take into account when testing the efficacy of vaccine candidates. So I would like to acknowledge the following institutions and individuals and collaborators, with a special thanks to Jomien Mouton, who is my MSc student, and here is a picture of him. And this work forms part of his MSc project. And I would also like to thank Validate Network. Although this work was not necessarily directly funded by them, my Validate Fellowship allowed me to be appointed at Stellenbosch University, which then allowed me to supervise my first two own MSc students, of which Jomien is one. And thank you for your attention.
Okay, so you're happy to take a few questions, Jomien?
Yes, I am.
So if anybody wants to ask a question, the easiest way to do it, along the top, there is the raise hand function. It looks like a high five. I've just done it now. So if anybody has any questions, just click on the raise hand and then Jomien can pick. So any questions for Jomien? I think you're off the hook. Oh no, there's one. Bernardo.
Sorry, Jomien. I'm a bit confused. Maybe you said it, but I missed it. How did you isolate the bacteria from the cured patients? Is it retrospectively, or what happened?
Sorry, I'm just trying to figure out. Can you hear me? Or am I on mute?
Yeah, I can hear you.
You can hear me. So these, sorry, that's maybe a good question. I apologize if it wasn't clear. So these were patients that were collected as part from a different study, and we actually have the sputum samples and the midgets available from these patients.
Right, so it was a retrospective study that you isolated. The bacteria were not necessarily isolated from the regions of where you could see the increased lesions or not.
No. So it was just from whole blood or sputum. Yes.
Thank you very much for that clarification. Thank you.
Pleasure.
Any other questions? You can just speak up as well if you're. Oh, hold on a second. We might have a. Oh yes, Helen.
Yeah, I've raised my hand. Is it okay for me to ask a question?
Yeah,
I really enjoyed your talk.
Go ahead, Helen.
Yeah, so it's about this sort of continuum between patients who become infected with a strain that is genetically more likely to give them persisters and a relapse. And then the other end of the continuum, you've got sort of patients for whom those genetic changes take place, you know, inside their bodies during the treatment. And I just wondered whether you could get a feeling for where you are on that continuum, maybe by some follow-up to have a look at the genotype, the sort of whether there was any isolates from those patient contacts, you know, the people who they basically caught the TB from, were those genetic changes present at the time that they caught the TB, you know, in their family members or the other people around them who had TB, or just to know really where it's sitting on that continuum, because obviously that's got implications for the sort of, you know, this sort of, if this is happening rather rapidly in in the body of people receiving treatment, then this is a sort of ongoing evolution where we might sort of develop a preponderance of TB strains that are more likely to be persisters. So you might really see a change over the years of the kind of TB strains that are circulating. So it's just having a feeling of how rapidly that might be happening, whether there's any existing sequence data that could help with that.
Yes, I think that's a very interesting point that you raised there. I think one of the things is that with persisters, it's generally been shown that it's not as a result of genetic mutations, and it's only recently that people are looking at genetic variations and how this contributes to persister formation. So I think starting off by looking at whether there are these present and how they contribute to this phenotype as a start. But I think to be able to track back and into generations and using data that's maybe available for that would be really nice. I think something in terms of a kind of like a phylogenetic analysis using samples would be nice for that. We are expanding our genetic analysis to specifically compare samples between the different phenotypes that we see and do phylogenetic analysis. And because we've got the genetic analysis for the bigger group of samples that we didn't test, we would be able to expand it to those for before and after treatment as well. But something else that we have available in our groups is also different lineages with distinct phylogenetic lineages, and these are also transformed with the reporters. So it would be interesting to see how genetic changes, for example, contribute to phenotype differences that we see there. I don't know if that answers your question.
Yeah, thank you. I mean, it does a bit, doesn't it? It's whether those polymorphisms that you saw were present when those patients became infected, and that's only really accessible if samples were taken from the other people that they were basically infected from at the time. Yeah, it's really interesting. I really enjoyed it, and I know that it's really new to be looking at genetics as a component for that.
Thank you.
There was one more question from Anthony. So I was wondering if we could quickly answer that question. I think we should probably then move on to Sam. Jomien, can you turn off your share? Because I think you're sharing your screen. I think that's you.
Oh.
You're sharing the team session we're all in anyway.
Sorry.
That's all right. And Anthony, if you could ask a question, and then we should move on to Sam's presentation. Thanks.
Thank you. That's a fascinating talk. Really enjoyed it. I think you touched upon in your last answer, but I was interested in the strains that you looked at and the lineages that they come from. Is there any sort of indication that the types of phenotypes that you see are linked with any of the phylogenetic lineages?
Anthony, I had some trouble hearing you. Are you referring to the PET-CT imaging?
I was referring to the strains that you sequenced.
You seem quite quiet, Anthony. It might help. Maybe type your question and then in the chat. Oh.
Might have to see that. Blakely, can you see the question? Sorry,
He just typed sorry. I think he's probably writing. I was interested in the strains. That's all he said. You said. The strains you sequence.
Yes.
Do you know their lineages?
Yes, we do. So we've done Spoligo analysis and RFLP analysis on these lineages, and interestingly, the 3 samples that demonstrated variants in were from the Beijing lineage. So yes, but that is something that we are definitely also looking at in a bit more depth.
I think then okay, thanks. That's Anthony says thanks. I think we should probably move on to Sam's presentation.
Yes.
Are you ready there, Sam?
I am, thanks, Blakely. So I'm just quickly switching on my video to say hi to everyone, but I will switch it off again so that you can focus on slides. And I'm about to share, Blakely, if you could just confirm that you can hear and see me and see the slide.
I can hear you. I can't see the slide. I can see Jomien for some reason. Hang on.
Sorry, I'm trying.
Can you? What's going on there? There we go. Ah, there we go. I can see the slides. Everyone else should be able to see the slides. Do let us know if you can't.
Okay, great. So I will go ahead. So good morning or afternoon, whatever is applicable here to you. Firstly, thanks to Blakely for organising the session today and to Validate for hosting these sessions, which I think are a great opportunity for people to connect. And sitting here, I've been very excited to see some familiar names coming up in the chat, people that I've worked with in the past, particularly in the UK. So hi. So I'm going to preface the talk by saying that similar to Jomien, what I'm presenting today is not work on the project that we are currently receiving Validate on priming for, but I hope to be able to share that work at a future meeting. So I'm hearing quite a lot of background noise. Okay, so as Jomien mentioned, this work is going to focus on PE and PPE proteins. So as most Validate members know, MTB is an intracellular pathogen, and initial contact with the host macrophage can lead to a spectrum of outcomes. And this in part is mediated by numerous secreted mycobacterial effectors, and one collection of such effectors falls within the PE and PPE families. So these are very large gene families. There are approximately 160 genes in total. And they are named for proline glutamic acid and proline glutamic acid motifs encoded at the start of the proteins. Both groups are characterized by this relatively conserved N-terminal region of about 110 amino acids and 180 amino acids in the PEs and PPEs respectively. Each subfamily is divided into 3 similar subgroups based on their more variable C-terminal domains. And the 2 largest subfamilies, both in size and number, are the PGRS and MPTR subfamilies. So these are the last abbreviations I'll use, I promise. So PGRS stands for polymorphic GC-rich sequence and MPTR for major polymorphic tandem repeat. And this refers to very GC-rich repeats that are found in the C-terminal regions of these proteins. So the PGRS and MPTR subfamilies are considered to be the most recently evolved and are only found in pathogenic mycobacteria, hence our particular interest in these. As many of you will know, PE and PPE secretion is mediated by the type 7 secretion system, which is also involved in the secretion of the well-known ESAT6 and CFP10 antigens. And so these, of course, have been used in the context of TB vaccines and diagnostics. Now, I'm clearly a sucker for punishment because these proteins are, difficult as they are a very long-standing research interest of mine, as some in the audience know. And over the years, we have studied their localization, genetic variation, regulation, evolution, and immunogenicity. So in work that is somewhat in line with Validate interests, I previously investigated the kinetics of immune responses to PE and PPE proteins in both bovine and human samples. And this work challenged the pervasive speculation that PE and PPE proteins represent a source of antigenic variation. it further identified novel antigens which could inform future vaccine or diagnostic development. And so PE and PPE proteins, along with Persister work, are an ongoing research focus within my group. So part of the work that we do has a much more applied focus, and that involves antigen discovery and characterization. But other work is much more fundamental. And in this, we are looking at how these proteins interface with and modulate host immune responses. And it is some of that work that I'm going to present to you today. So MTB, as you know, is adept at subverting host macrophage defense mechanisms, and PE and PPE proteins have been shown to play a role in several steps here. So, for example, some family members can inhibit phagocytosis, while others bind to toll-like receptors and influence downstream signaling. Other family members have been shown to be implicated in the inhibition of production of reactive nitrogen species, and others again have been shown to interfere with phagosome maturation. And so there's therefore extensive evidence that these proteins are important factors contributing to MTB pathogenesis at the level of the innate immune response. So as mentioned previously, the most well-studied subset of the proteins is the PGRS family. And together with the PPE and PTR proteins are a particular interest within our group. So one of the several difficulties in studying these proteins is that there are so many of them. And even in an easier organism than MTB, it would be somewhat challenging to systematically knock them out. And even then, seeing a phenotype might be unlikely. However, we are very fortunate to have a generous collaborator in Wilbert Bitter from the VU Amsterdam in the Netherlands. And his group had developed a mutant strain defective for PGRS and MPTR secretion. So some background to this: in a series of studies in Mycobacterium marinum and Mycobacterium tuberculosis, Wilbert's lab has demonstrated that deletion of a particular PPE gene, PPE38, abrogates secretion of PGRS and MPTR proteins. So although PGRS proteins are still produced, as you can see in the, they are not secreted in strains that are lacking an intact PPE38 gene. So of great interest is the finding that strains defective for PGRS secretion are hypervirulent. And so this has been shown both in Mycobacterium marinum using a zebrafish model, as well as for Mycobacterium tuberculosis in a mouse model. It has also been shown that clinical isolates of Mycobacterium tuberculosis with natural PPE38 disruptions have defective PGRS and MPTR secretion. So interestingly, PPE38 deletions and disruptions are common in successful modern MTB lineages, such as the Beijing lineage. So initially, this finding seems counterintuitive because we talk about PEs and PPEs as virulence factors, and when they are absent, the strains are hypervirulent. So we set out to investigate the mechanisms underlying these findings of hypervirulence associated with reduced PGRS secretion. And to do this, we used the THP1 human macrophage cell line. We infected this with a wild-type strain, that is CDC 1551, which secretes PGRS and MPTR proteins, with a PPE38 mutant, which does not, and a strain in which the secretion is complemented. So following these infections, whole cell proteomics were performed to probe host responses. And we did this using both label-free and pulsed SciLac proteomics, but in the interest of time, I will just talk about the former. So the findings from the proteomics were validated using a range of approaches. And I just need to pause for a moment and emphasize that when I say we, I mean that this work was largely driven by a very talented PhD student in my group, James Gallant. So very briefly, for the label-free proteomics, THP1 macrophages were infected with the wild-type, deletion mutant, and complemented strains, and then harvested at 4 hours and 18 hours post-infection. Following lysis and proteome purification, trypsin digestion was performed, and the tryptic mix was analyzed by mass spectrometry, followed by data analysis to identify the differentially expressed proteins. As mentioned, proteomes are characterized both at 4 hours and 18 hours post-infection. And this revealed that there was little change at 4 hours, as we would expect. But by 18 hours, there were 39 proteins that were down-regulated in the mutant infected macrophages and 11 that were up. So within these proteins, we observed a general trend towards a pro-inflammatory response in wild-type infected macrophages compared to an anti-inflammatory response in macrophages that were infected with the PGRS and MPTR secretion deficient mutant. So zooming in on key proteins that were differentially abundant in the two sample sets, shown here are results for IL-1 beta, a pro-inflammatory cytokine, Nfkb2, DDX58, which is an innate immune cytoplasmic sensor, and ISG15, an interferon stimulated gene. So we note that there was upregulation of all of these in the wild type relative to the mutant infected macrophages, and this is indicative of both altered interleukin and interferon responses. Furthermore, gene set enrichment analysis of the differential response between the wild type and mutant affected macrophages revealed that Nfkb signaling was the most enriched pathway. So very broadly, Nfkb signaling triggers a pro-inflammatory response. Based on this and other proteomic signatures that we saw, we questioned whether the deletion mutant was modulating macrophage polarization. So just a little bit of background on this. I'm showing a very simplified view of macrophage polarization, and I apologize to immunologists in the audience. But this is just a framework for what we subsequently looked at. So it's well known that macrophages demonstrate phenotypic heterogeneity. And depending on the signals they encounter, they can be differentially activated into a so-called classically activated or pro-inflammatory state known as M1 macrophages, or into an alternatively activated anti-inflammatory state, or M2. So these populations are defined by distinct surface markers and secreted cytokine profiles. And as one would expect, M1 macrophages secrete pro-inflammatory cytokines and are considered restrictive for MTB, whereas M2, by contrast, are considered permissive. So interestingly, it's been shown that levels of M2 macrophages increase over the course of TB infection, and it's further been predicted that the M1-M2 ratio may drive granuloma outcome. So therefore, this hint that we saw from the proteomic data that the PPE38 deletion may be modulating macrophages towards an M2 phenotype may provide some clues as to the virulence of the strain. So we were therefore keen to follow up these findings. So to do this, we performed ELISAs on macrophage culture supernatants at 48 hours post-infection. Here we looked at IL-12P70 as a proxy for an M1 phenotype and IL-13 as a proxy for M2. And indeed what we saw was that IL-12P70 was upregulated in wild type relative to the deletion strain, and conversely IL-13 was down in wild type relative to the deletion strain. So further characterization of this does need to be done, but it does support the idea that the PPE38 mutant elicits an anti-inflammatory permissive M2 phenotype. So as previously mentioned, the Nfkb signaling pathway is a key player in triggering inflammatory responses. So Nfkb activation can occur through two major pathways, namely the canonical or REL-A mediated pathway and the non-canonical or REL-B mediated pathway. In the former, an Nfkb precursor protein, P105, is processed and binds to REL-A prior to translocation of a P50 complex to the nucleus. And this triggers inflammatory gene transcription, a burst of this. So this major pathway can be supplemented by the non-canonical pathway, in which a P52 REL-B subunit or complex undergoes nuclear translocation. So we therefore probe this in a bit more detail using a combination of Western blotting and confocal microscopy. So from the Western blotting data, we see that the Nfkb P50 subunit is available for binding in all conditions, in all macrophages infected with any of the strains. However, an inhibitory P100 subunit is upregulated in the wild-type infected macrophages only. And we see, along with this downstream upregulation of the downstream effectors, ISG15 and IL-1 beta. We therefore expect that the wild-type infected macrophages follow this canonical pathway. But based on these results, it was a bit more difficult to understand what pathway was operational in the mutant-infected macrophages. So, of course, this is all done on whole cell lysates. So we wanted to look a bit more closely at localization, and we therefore performed confocal microscopy. So bear with me, I'll talk you through this. So very briefly, what we see is that there is nuclear translocation of REL-A in macrophages infected with the wild-type strain, but not the deletion strain. And this is as predicted. It provides support that the canonical pathway is operational in wild-type infected macrophages. So, although we do see translocation of REL-B in the mutant-infected macrophages, there is no associated P52 translocation, which we would expect if it was the non-canonical pathway. But what we do see is P50 translocation. So now this is getting a little confusing because it seems that instead of seeing REL-B P50, REL-A P50, we see REL-B P50. But this then suggests that something else is happening besides these classical pathways. And actually, a little bit of probing of the literature indicates that this REL-B P50 complex may actually be involved in anti-inflammatory responses, which fits into this emerging picture. So very briefly, to sum up what we think is happening, it seems that reduced PGRS and MPTR secretion leads to a dampened inflammatory response. And the combination of our proteomic, Western blotting, and confocal microscopy data suggests that the translocation of the P50 REL-B complex might act as a molecular switch, resulting in the skewing of macrophages towards an M2 permissive phenotype. So therefore, strains with reduced MPTR and PGRS secretion due to a genetic mutation may therefore skew macrophages towards a more permissive environment, allowing MTB to establish infection and multiply. So we know that many successful lineage 2 or Beijing strains have this naturally reduced PGRS and MPTR secretion, and this could be a contributor to the success of these strains. In other strains where they retain the natural ability to secrete PGRS and MPTR proteins, this secretion may be differentially regulated at different points in infection, and this can fine-tune the environment of the bacteria. So admittedly, there's a lot to still do to confirm and build on these findings, and a particular interest moving forward is what, if any, are the host targets of these intriguing PE and PPE proteins. So of course, if there are any Validate members with experience in doing this for effectors and other pathogens, or ideas or questions about the PE, PPE proteins, we'd love to hear from you. So I hope that the two talks today have given a bit of insight into the type of work we do in our little corner of the TB world, and we are always open to new collaborations. So with that, I'd like to acknowledge my funders, collaborators, my fantastic team at Stellenbosch University, and of course, Validate for the opportunity to speak to you today.
Thank you very much. Does anybody have any questions? Again, you can raise your hands, or you can just go on and unmute and speak. So, any questions? You can alternatively type your questions into the chat if you're having some connectivity issues. You can raise your hand, although I think if you just unmute and ask your question, that'll work as well. Alvaro, do you want to go ahead?
Hello, yes. Thanks for this fascinating seminar. I'm not a microbiologist and the work with tuberculosis, but I found this very fascinating. I wanted to ask you whether after secretion of these proteins, they get further processed like a proteolyzed, so they can target several pathways that activate macrophages.
Yes, that's a great question. So it's hard to answer as a general statement. So as I've talked about, there are different subsets within the PE and PPE protein families, and we know that upon their secretion, some of them are differentially cleaved. So we know at least the Mycobacterium itself cleaves them differently. In terms of what happens in the host, it is pretty much an unknown. There is some indication or older literature that indicates that the PGRS proteins in particular may interfere with proteasome processing. And the thinking at the time was that this could interfere with antigen presentation. But again, this has not been extensively followed up as far as I'm aware.
Thank you.
Susie.
Yes, hello. Hi, Sam. That was a great talk. Really interesting. I just wondered with the IL-1 beta, if there's a follow-on effect with apoptosis or necrosis. Sorry.
Hi, Susie. Nice to hear your voice. Great question. And actually, there's a very recent paper that's come out looking at particular PE/PPE family members and their effect on apoptosis. And it's not something that we've looked at, but it is definitely something to do in terms of follow-up. There is other work, older work again, that's indicated that there may be an influence of certain family members on apoptosis and necrosis. So it's a great question, but linking that with IL-1 beta, it's something on the to-do list.
Oh, great. Thank you. Cheers.
Adelaide, would you like to ask a question?
Okay, I'm seeing a question in the chat, and I'll just repeat the question if anyone can't see it. So thanks for the question. It says, do you think the proteomic and immunological changes you see could be time dependent as in duration of infection? Do you expect these trends to be different if the macrophages were infected for longer than 18 and 48 hours? So it's a great question. We haven't prolonged the proteomic analyses beyond 48 hours at this point. Part of the concern there is the technical issues in that what we're seeing the initial uptake and engagement of the macrophages. And so I think with longer infection times, we would have to control for cell lysis and reuptake. And that's a question that we've had from reviewers in the past. So at this moment, the simple answer is no, we haven't done extensive time courses, but it certainly is something to think about. And I think to remember that, the immune response is not just the macrophages, so what else happens downstream? So it's a great question, thank you.
There was another question from Theolis, who asked, the hypervirulence phenotype is associated with increased proliferation, ?
Apologies, I missed that. So no, so at least in the experiments that we've seen in our work, the phenotypic differences we've looked at uptake, replication of the MTB in the model that we're using, and there's no difference. In terms of the mouse work, I'm going to hit a complete blank here and say that I think there might be, and I'm very embarrassed to say I'll have to look that up for you. So.
And from Adelaide as well, there's a question there, which I can't actually see.
Okay,
because
I can see it. The question is, in terms of dynamics, what is it? Oops, things keep popping.
Yeah, same with me.
I just have to keep killing those. It's like a game. In terms of dynamics, what is your thoughts of the relevance of this mechanism in the context of early versus later stages of infection and disease? I think that really, again, is an excellent question. And it's a little bit what I was alluding to in the closing of the talk. So we know that at least for those strains that secrete PGRS and MPTR proteins at all, like some of the Beijing lineage strains, we believe that that is linked to this increased ability to take hold in terms of the infection and so on. But other strains we know do secrete PGRS and MPTR proteins, but we know at least from transcription data and from proteomic data over a time course of infection in animal models, there's differential expression over time. And so what I think is very intriguing is the bacteria able to regulate this expression to fine-tune its environment? So, you know, if it shuts them down, does it make this a more permissive environment, which may be beneficial at some stage, but at other stages you might want to have this more pro-inflammatory response.
There's a couple more in the
wow, okay.
Yeah. Do you think the PE/PPE proteins have a role in inducing innate trained immunity? And that's from Fabiana.
Wow, I'm going to have to say I don't know. That's an I don't know to that one.
And there's another one from an anonymous guest. In terms of PE/PPE proteins in Mycobacterium pathogenesis, what do you think about that, I guess?
Okay. Okay, that's quite a broad question, but maybe just coming back to the introduction of the talk, as I tried to indicate, there are numerous reports looking at different members of the PE and PPE family, and I think my view is that the different subsets play different roles. And so it may be that the PGRS and MPTR proteins are involved in this molecular switch that we've described, but others may have a completely different role. But in terms of their evolution, one thing we know is that the PGRS and MPTR are the most recently evolved, and they are only found in pathogenic mycobacteria, whereas some of the other family members are found in non-pathogenic mycobacteria as well. So I think it's really important, if I leave you with one message, they are not all the same, and they are probably playing different roles.